In this video, I'm going to talk about the streak plate method. So the streak plate method is a method for isolating microorganisms from a mixed culture. We can do a streak plate method from an agar plate, or we can do a streak plate method from a broth. So I'm choosing to do streak plate method from a broth. So let's pretend that we just swabbed a patient's wound, and in that sample we see a whole bunch of different types of cells and all different types of bacterial cells mixed together. We were to take that patient's swab, we'll slosh it around in a tube, it gets really cloudy, and now we can take this broth and we can smear it on the plate thinner and thinner and thinner until we isolate colonies. A colony is an individual separated growth of bacteria. Typically, a colony arises from the cell divisions of one bacterial cell. So you have one bacterial cell that has landed on the plate, divided, and grown into this visible cluster of bacteria. It typically takes 2 days or so before you can actually see the colony. So when you initially put it down on the plate, it's just one cell, it's microscopic, but then it'll grow to this visible cluster of bacteria. All of the cells inside of a colony are essentially genetically identical because they should have arisen from the cell divisions of one bacterial cell. And that makes it a really useful diagnostic tool. Then we can look at individual colonies and start to decide which one looks weird, which one looks normal, and which one do we want to pay a little closer attention to in order to diagnose the infection. So to do our streak plate method, we want to start by labeling our plate. And we're going to label it with the information that we have. So I happen to be culturing from an E. coli culture tube, so I'm just going to write E. coli. But if this were a patient sample, it would be labeled with that patient information. And then I'm also going to put today's date. I am going to pick up the loop, and I'm going to flame sterilize the loop. And then let it cool off. I'm going to grab the bacterial tube, aseptically take off the cap, and I'm going to swirl it around to obtain a loop full of bacteria. I'm going to put the cap back down and set down the tube. Then I'm going to transfer bacteria to the original section on the plate. So I'm going to just lift up the lid slightly, and I'm going to smear it in one section of the plate. Some people like to physically draw quadrants on the bottom of their plate. I like to just rub it in sectors along the edge. So now I'm going to flame sterilize again. Now the goal of this is to take the bacteria that's on the plate and spread it thinner and thinner and thinner until you get those isolated colonies. So I'm going to rotate the plate 90 degrees, and then I'm going to smear from the first segment into the second segment. And I'm going to grab it about 3 times and smear it around in that new segment of the plate. And I'm going to flame sterilize the loop again. I'm going to rotate the plate another 90 degrees, and then I'm going to stab the loop into the side of the agar so that it cools off the loop, and I'm going to spread it from the second quadrant into the third quadrant. And again, I'm going to take it 3 times. I'm going to flame sterilize the loop. And then I'm going to rotate the plate 90 degrees. I'm going to stab this into the side of the agar, and then I'm going to rub it from the third quadrant into the fourth quadrant. You'll notice when I was doing this transfer that I only opened the top of the plate enough to get the loop in there. I also made sure not to take the plate off of the countertop. I left the plate on the countertop. That's the best way to do the streak plate method. Sometimes I'll look into a clinical lab and I'll see people holding the plate up like this and going in there like that. Not only are you contaminating it with your breath at that point, but you're risking dropping it on the countertop. So it's best to leave it on the counter when you're performing your streak plate method. The other way to do it is with the quadrant method. So I'm going to show that method now. You again want to start by labeling your plate. And then you can break the plate down into visible quadrants. To do this, you're going to flame sterilize your loop, obtain the bacteria, aseptically take off the cap, swirl it around in the tube, put the cap back on, and then you're going to smear the bacteria in the first quadrant. Because I drew the quadrants on the bottom of the plate, I can see through the plate where the quadrants are. So now I know I just streaked into that quadrant. I'm going to flame sterilize. And then I'm going to rotate the plate, cool off my needle, and spread it from the first quadrant to the second quadrant. And I'm going to smear it 3 times back and forth. Rotate the plate again, flame sterilize my loop, cool it off, then from the second quadrant to the third, rotate the plate again, and cool it off, and then from the third quadrant to the fourth. Again, the goal is to put some bacteria on the plate and then smear it thinner and thinner and thinner until we can have single cells landing on the plate. When that happens, those single cells are then going to grow into a larger cluster, a colony, and then we'll be able to evaluate that colony. So this plate was put in the incubator for a couple days, so about 48 hours we keep it in the incubator to grow. It grew at 37 degrees Celsius, which is 98.6 degrees Fahrenheit, or body temperature. This is E. coli growing on the plate, and you can see how there are now isolated colonies. So looking at the plate more closely, you can see that this is the original quadrant that the bacteria was put in. It was streaked into this quadrant, and then streaked into this quadrant, and then finally into this quadrant. Each one of these little dots is a colony. So now we have isolated colonies. The other thing that you can notice about this plate is all of the isolated colonies are the same color, approximately the same size, the same shape. So now we can confirm that this was a pure culture, or this was only one species that is growing on the plate. If you're going to culture from a patient, usually you're going to see lots of different colors, and there's going to be a lot of different species of organisms that are growing from a patient sample. Unless it's going to be cultured from what we know as a sterile site, cerebral spinal fluid should probably only have one microorganism in it if the patient has meningitis. If they have multiple microorganisms in there, then they're probably in really bad shape. So this is what isolation looks like. So you can see big cluster, smaller cluster, and here's where we can start evaluating individual colonies. Anytime we want to do a workup on a microorganism, we're going to want to work up a single colony, and that's what we can run all of our tests on. So we would take our loop and we would scrape off that one colony and do some follow-up work on that one colony.